Most of our life, we've been associating the color green to a healthy environment. When you go to the mountains and you see a scenery with trees vibrant with the color green, you will feel clean and revitalized as green means life. But what if I tell you that green is also associated with pollution and death? So that's what we will be talking about in this more or less 40 minutes of discussion. Specifically, my groupmates and I will be sharing what we've learned about the trophic states and eutrophication. First, you may ask, what are trophic states? The word trophic is derived from the Greek word trophy, meaning nourishment or nutrients. Therefore, trophic states can be described as the condition of different water bodies, like lakes and rivers, based on the amount of nutrients present in them. To properly classify the water bodies depending on the amount of nutrients present in them, the Trophic States Index or TSI was designed by limnologists. The most commonly used formula of the TSI was created by Robert Carlson, wherein three important variables were used. TP for total phosphorus, since phosphorus is a limiting nutrient, meaning it is one of the least present nutrients that an organism like algae needs to grow. Therefore, an increase in phosphorus controls the algal growth. Ca for chlorophyll A, since it is the color pigment found in algae. The more chlorophyll corresponds to more phytoplanktons and more eutrophic state of the lake. Lastly, Sd for sucky depth, as it is used to measure the transparency of water, meaning the lower the transparency, the denser the algal populations and other suspended solids in the water. Depending on the TSI value you will obtain, a water body may be classified as either oligotrophic, with a less than 40 TSI value, mesotrophic, 40 to 50, and eutrophic, more than 50 TSI value. An oligotrophic water is clear and is rich with oxygen, while a moderately clear water can be observed in mesotrophic water. Eutrophic then are opaque with a possible algal bloom and ultimately death of fishes. In Greek, eutrophication means the process where a body of water becomes well-nourished. But in the sense of limnology, it's more likely an overnourished water body. The process of eutrophication can be both natural and human-induced. Natural eutrophication, where the basin gradually fills in with nutrients and sediments, that occurs over long periods of time. However, humans, by altering nutrient inputs through various land use practices, have greatly increased the pace at which eutrophication can occur. This cultural eutrophication occurs through a timely process. First, excessive nutrients like phosphorus from fertilizers are flushed from lands into rivers or lakes by rainwater. These pollutants induce the growth of algae and other plants. Algae then blooms in a layer preventing sunlight reaching other aquatic plants. They then die and oxygen in the water is depleted. Dead plants are then broken down by their composers, consequently using up even more oxygen. Oxygen levels then is depleted to a point where death impends upon fishes and other organisms. Keeping in mind all the things I've discussed, we are now to look into 3 case studies to better understand in detail eutrophication in the Philippine setting. But before that, I will show you global examples of eutrophic inland waters. What you have to do is to guess what lake or river am I showing to unlock our case studies. But don't worry, I will give you a clue as to which country it belongs. So let's start. This Canadian lake had a giant algae bloom took over on the summer of 2014. It was so big that you could see it from space. The algal bloom kept coming back and eventually, people pushed for the lake to have the same legal rights as humans, where it recognizes the lake's right to exist, flourish, and naturally evolve. Do you have any idea what am I talking about? Yes, Keith, you were raising your hand.
Is it Lake Erie?
Yes, you're right, Keith. So the Lake Erie Bill of Rights I am talking about recognizes the lake's right to file a lawsuit if it's been harmed. It may sound ridiculous, but it really took another step to nature's conservation. So with that, you have unlocked the first location for our case study, and it is the San Cristobal Subcatchment. On to our next lake is known as the third largest lake in China. In this picture in 2019, the algae-infested waters are clearly visible. In 2007, the algal blooms were so severe that the outbreak was declared a health emergency. Water supplies were suspended, leaving 2 million residents without drinking water for several weeks. What famous lake is this? Okay, Pauline, I see you raising your hand.
Based on what I remember po, it's Lake Tai.
Correct, Pauchen. That's a retentive memory you got there. So, algae blooms in Lake Tai have been reported in the lake since the 1980s. Many attempts have been made to salvage its water quality. However, the lake remains to be highly polluted. After San Cristobal Subcatchment, we will virtually go to Silang Santa Rosa Watershed as our second case study. On to our last inland water, a river this time. This river is located in the United States and is known as the longest river of North America that ends in the Gulf of Mexico. The Gulf has a massive patch of hypoxic water that forms every summer. This patch is called a dead zone since aquatic organisms will not be able to survive if they reside in this patch. Does this river sound familiar to you guys? So Mimi's raising her hand. Do you have any idea what it is?
Yes, it is the Mississippi River.
Precisely. So, the problem of this dead zone starts thousands of miles away when some fertilizers applied to farmland get washed into the Mississippi River, where it induced the growth of algae. And with that, you have unlocked our last, but of course not the least destination, the Marikina Watershed. So, a very well done for those who participated. You might now be the next limnologist who will solve all this eutrophication around the world. Now that we've unlocked all our 3 locations, let us go to the first one, which is the San Cristobal Subcatchment of Laguna de Bay. This place was studied by Jojejole et al. In 2021, wherein they examined the effects of the industrial zone on the physicochemical and biological conditions of the San Cristobal and Diezmo River. To do this, they compared the upstream sites located before the manufacturing plants and the downstream sites near the industrial outlets.
So to start off, for us to discuss this issue, let us play a game called 4 Picks, 1 Word. So, first one, based on these photos, what scenario can you observe? Yes, Aki?
Based on the image, we can see that there is a factory beside the bodies of water.
Yes, that is correct. So, what do you think is the effect of these factories to the surrounding water?
Well, because of the waste materials that are being released through these factories, the water quality can be greatly affected since it can be polluted, which in turn can disrupt the organisms living in it.
Yes, and that is what is exactly happening in this landscape. And yes, there has been several studies have demonstrated the correlation between industrial zones and water pollution, where declining water quality serve as a consequence of a rapid economic growth and industrialization. And in fact, there has been an increasing concern in developing nations in Asia where most industries discharge waste directly into rivers without proper treatment due to limited environmental regulations. Now, let's proceed to the next photos. 4 Picks, 1 Word. Okay, so what do you think is shown in these photos? Are you familiar with these photos? Okay, yes, James.
Yes, I think I'm familiar with the area. Is it Laguna de Bay?
Yes, that is in fact Laguna de Bay and its catchment, which includes the San Cristobal Subcatchment. And in the last 30 years, it has undergone rapid land cover change, where rural areas have been developed into settlements and industrial areas, which deteriorated river quality in terms of these properties. So next photos. Are these organisms familiar to you? So if yes, can you tell me what it is and what is its importance in studying freshwater biology? Okay, yes, Eunice?
The organisms in the photo are benthic macroinvertebrates, and based on what I know, they can serve as bioindicators for the quality of the body of water.
Yes, that is correct. Thank you, Eunice. And since disturbances in water are brought about by human activities, it mainly affects the assemblages of river biota and its group of bioindicators, the benthic macroinvertebrates. So, this group offers a set of distinct advantages as bioindicators. So, in particular, their ubiquity in sedentary and its longer life cycles, constant exposure to varying water quality conditions, and it's easily identified and collected that made them an efficient indicator. So in this talk, we will tackle the effects of an industrial zone located at the downstream site of San Cristobal Subcatchment on water quality and benthic macroinvertebrate communities. For our next item, what do you think is being referred in these photos? Oh yes, Tori?
I think those are the equipment that we use in testing water properties.
Yes, that is correct, Tori. Thank you. So, to be able to assess the spatial and temporal variability in water quality, the researchers of this study used these equipment to measure the specific properties of dissolved oxygen, temperature, conductivity, and pH. Ammonia, nitrogen, and phosphate phosphorus were analyzed through portable colorimeter kits, while nitrate nitrogen was through a photometer. So, based on what we've studied in this course, since they also collected microinvertebrates, what do you think is the metric that is being referred based on the 4 pictures in this slide? Yes, Alfred?
I think it's the Ephemeroptera, Plecoptera, Trichoptera, or EPT richness, which we use in water assessment quality.
Yes, Alfred, that is correct. And aside from the richness metrics, abundance, taxon richness, Chironomidae richness, and Shannon-Wiener diversity index, this was also calculated. Now, for the results of the study, let's play another game. So I will show you the figures and tables, and we will determine together the relationship of each water parameter and its interaction with macroinvertebrates and varying seasonality and location. First, let's look at the temperature. So, based on the values, what do you think? Is there a significant difference, or there is no significant difference in both the upstream and downstream and the wet and dry season? Okay, Keith?
Well, based on the ANOVA values when it comes to temperature, I don't think there's a significant difference in both location and seasonality.
That is correct. Thank you, Keith. So it can be observed that there are only narrow ranges for an average temperature and pH that were noted in both reaches of the stream and during the wet and dry season. So, how about the average DO of the upstream versus the downstream? Which one has the higher dissolved oxygen? And why do you think so? Okay, again, Keith?
Well, by looking at the table, maybe as we go from upstream to downstream, we can observe a difference in DO.
Again, that is correct. Okay, now let's move on with our next parameters. Our 5-day biochemical oxygen demand, ammonium nitrogen, nitrate nitrogen, phosphate phosphorus, and total suspended solids. So, what can you observe in terms of the trends of these parameters according to its location, from upstream to downstream? Yes, Alfred?
Let me take a look first. Okay, so based on the table, all of the parameters from BOD, ammonia nitrogen, nitrate nitrogen, phosphate, phosphorus, and TSS increases from downstream to upstream.
Thank you for answering. And there is an increasing spatial trend from upstream to downstream that was observed for BOD, ammonia nitrogen, nitrate nitrogen, phosphate phosphorus, and TSS. And in fact, these values in the downstream site has exceeded Philippine water quality guidelines for BOD5, ammonium nitrogen, phosphate phosphorus, and below the guidelines for dissolved oxygen. In addition, Peralta et al. Found a distinct spatial variations with highest concentration of ammonium and total phosphorus and reduced DO level in the streams located in built-up and industrial areas. So, what about the BOD5, nitrate, and total suspended solid trend comparing dry season? Is it increasing or decreasing? Yes, Asi?
I think that BOD5, nitrate, and TSS are greater or increasing in wet season compared to dry season.
Okay, how about the phosphate phosphorus amount comparing wet and dry season?
Decreasing during wet season.
So thank you for that, Asi. So this goes the same from upstream to downstream. And in addition, the conductivity measured during the dry season revealed a higher conductivity values and variability downstream. Thus, the analysis reveals the collinearity that exists between dissolved oxygen concentration and nutrients ammonium to nitrogen and phosphate to phosphorus, as well as the total suspended solids, indicating their collinearity, higher dissolved oxygen values upstream and lower dissolved oxygen values in the downstream areas. Now let's discuss the benthic macroinvertebrate communities of the San Cristobal Subcatchment. For our first question, which season do you think has more macroinvertebrate individuals collected? Is it the rainy season or the dry season? And why do you think so? Yes, Angel?
Based on the data obtained in the study, I think there were more individuals observed during the rainy season than the dry season. Maybe because there are high amounts of water compared during the dry season.
Thank you for that, Angel. And yes, higher abundance, family richness, and diversity were observed during the rainy season. And they are in contrast with the results of other studies, where abundance and taxon richness decrease with increasing rainfall, or no temporal difference at all. However, as you can see, rainfall as a disturbance is absent. Benthic macroinvertebrates could further proliferate during the rainy season. Why is that so? So, the amount of rainfall might not be enough to remove the substrates or the benthos. Now, let's talk about the spatial abundance. Are there more benthic macroinvertebrates upstream or downstream?
Well, based on the data, I think that benthic macroinvertebrates are more abundant in the upstream compared to the downstream, since upstream areas are comparably undisturbed.
That is correct. So, moving on. With the proportion of the major benthic macroinvertebrate taxa of the San Cristobal Subcatchment, which of these families dominated the downstream area?
Is it the insect Chironomidae po?
Again, that is correct. And as you can see, the families belonging to the EPT in its abundance proportion declined from upstream to downstream area. So this is accompanied by the increase of Chironomidae abundance and proportion, which can be attributed to the organic pollution because of anthropogenic sources. These pollution-tolerant families clustered in the downstream sites with high values of ammonia and phosphate. Now that we've seen where the macroinvertebrate families are found and variations in properties of both location and season, let's sum it all up. Presenting here is the redundancy analysis ordination diagram for benthic macroinvertebrates and water quality during the dry and rainy season. So the results of RDA show the variation of benthic macroinvertebrate community structure has a strong relationship with water quality variables. So as seen in this graph, dissolved oxygen concentration is positively influenced all the response variables except the Chironomidae richness and proportion, which is negatively affected. And on the other hand, the nitrate nitrogen, pH, and temperature have no profound influence in the benthic macroinvertebrate communities. And finally, based on the observed pattern in the study, variations in water quality variables were more pronounced between sites rather than in between seasons. And that's it for our first stop here in San Cristobal Subcatchment of Laguna de Bai. I hope you understood the tropical river and stream system changes in response to industrial land use. So, see you on our next stop!
Now we made it to our next stop, the Silang Santa Rosa Watershed. First, can you tell me what differences you can see here between these three pictures? You may describe anything that's within its surroundings like the color or anything that you can notice. Any show of hands? Oh yes, James.
Yung picture sa left side, yung pinaka-greenest, pinaka-greenest pa. Well, what I mean to say is, evident yung pagka-green or yung vegetation sa yung nasa left side compared dun sa nasa right side, na parang mas urbanized kasi nga mas maraming buildings and mga bagay. Kumbaga, alam natin na mas polluted siya ganun. Yung nasa middle naman, parang it's more of a mix between the two kasi may parts na green siya, vegetated, tapos may mga parts din na urbanized, yung maraming buildings.
Kuha! So James' answer basically sums up the stream system of the Silang Santa Rosa Watershed or SSRS. To give you a brief background, SSRS is an urbanized tropical stream system situated at the west portion of Laguna de Bai, being covered by vegetated and built-up areas. So the three pictures I presented earlier were the upstream portion, comprised mostly of forested land now being put into cultivations, the midstream portion where sugarcane plantations and industrial and residential land conversions are starting, and the downstream portion, which is the most urbanized part, comprised mostly of settlements harboring little to no vegetation. It is also worth noting that SSRS to date has a poor installation of wastewater treatment plants. Provided this information, we can say that a lot is actually changing within the area, leading to pollution and eutrophication as rapid economic growth took place. This is where the term Southern Urban Hydrosystem Syndrome, or SUS as Swanson et al. Defined it, comes in, which tackles the geophysical, chemical, and ecosocial aspects that drive urban hydrosystem restoration. That is why a recent study by Peralta et al. In 2020 assessed several sites in these areas as shown in this sampling map, where they focused on discriminating the co-varying effects of nutrient pollution and deforestation on benthic macroinvertebrate communities. Their study tested different environmental variables but focused on assessing the measured amounts of nutrients such as the total phosphorus, or TP, dissolved oxygen, or DO, nitrates, and ammonia. They have also gathered Geographic Information System, or GIS variables, including HPD and the catchment of each monitoring site, collected and identified benthic macroinvertebrates, and lastly used statistical analysis to examine how these variables co-vary or are interconnected with each other in inferring now the trophic state of SSRS. Among the environmental variables, the first prominent factor that contributes to eutrophication in SSRS is the total phosphorus or TP concentrations, wherein it has shown a clear spatial variation between the upstream portion, which had values that are less than the detection limit, while many sites in the downstream portion had values above the upper limit of hypertrophication. Moreover, increasing values of TDS, salinity, and conductivity were also observed with increasing TP concentration. DO, on the other hand, decreased significantly with increasing TP concentrations. And surprisingly, nitrate concentrations also decreased with increasing HPD and fraction of built-up areas. Provided the background on the portions of SSRS I've mentioned earlier, what do you think is the main contributor to the increase in TP loadings as we go downstream? Is it letter A, domestic waste, or letter B, agricultural activities? Yes, Matthew?
Hello po, ang sagot ko po is letter A, domestic waste.
Yes, tama. Their results show that HPD, or human population density, had the most influence on the total phosphorus loadings in the stream, as this is attributed to the residents' way of living, where they usually pollute streams with domestic waste such as septic and phosphorus-rich detergent waste, while industrial establishments also contribute by deforestation and directly discharging their wastes. This then leads to the excessive primary productivity or growth of algae, leading to depleted oxygen or hypoxia, thereby explaining the significant decrease of DO with increasing TP. The other determining factors of human activity and trophic state in SSRS are nitrates and ammonia. The unusual decrease in nitrate concentrations with increasing HPD and fraction of built-up areas can also be explained by the effects of hypoxia, as this process is known to inhibit nitrification and may enhance denitrification, which results in the removal of nitrogen from stream ecosystems. These anomalies strengthen the argument that TP is really a better indicator of nutrient loadings in urbanized stream systems lacking wastewater treatment plants such as the SSRS. In connection to the environmental parameters, we now again have here our representative bioindicators, which are the benthic macroinvertebrates. Across all sampling sites, stream benthic macroinvertebrates from 36 genera were collected and were largely dominated by Diptera, followed by Ephemeroptera, Coleoptera, Trichoptera, and Odonata, while the remaining were rare taxa. Based on the choices and clues provided here on the screen, which do you think would thrive in the upstream portion of the Silang Santa Rosa Watershed? Yes, Manila.
Is it A po ba, yung mayflies and caddisflies? Tama po ba? Kasi sabi ni Anne kanina, sa downstream daw yung chironomids eh.
Hindi lang naman yun yung reason, pero tama ka naman, Manila. So the results I've just shown you can be easily explained in this PCCA triplot, which basically exhibits that sites 1 to 7, or the upstream part which had higher DO and nitrates, and lower conductivity, ammonia, TP, and canopy openness, were positively correlated with these macroinvertebrates, many of which are mayflies and caddisflies from Ephemeroptera and Trichoptera. These taxa are known to be oligotrophic and mesotrophic, hence having high abundance in areas with higher DO concentration. In sites 8 to 13, on the other hand, which are comprised of the midstream and downstream portion having the opposite condition from the former, was tolerated by oligochaetes and chironomids, which are hypoxia tolerant. These explain that pO is the most critical factor controlling macroinvertebrate assemblages. Lastly, a factor that also contributes to assessing the trophic state or eutrophication in the SSRS through macroinvertebrates is the presence of diverse functional feeding groups associated with canopy openness. This includes collector-gatherers, shredders, and filterers that depend on allochthonous food sources from riparian forests. In the case of SSRS, deforestation in the riparian areas led to high canopy openness, thereby providing algal growth due to the strong light intensity. This allowed for eutrophic algal grazers to thrive, providing negative impacts on the stream's EPT index. The co-varying effects of nutrient pollution with other environmental variables, anthropogenic factors, and urbanization clearly added to the current eutrophic state of the SSRS. It is clear that the stream is in dire need of rehabilitation, not only by means of river solid waste cleanup projects, but by means of rehabilitating its riparian vegetation, given that until today, it is still considered an unprotected area. So that is it for us here in the Silang Santa Rosa Subwatershed. You're now on to your next stop, the Marikina Watershed.
We've come all this way, and at long last, we've reached our final destination, the Marikina Watershed. But let me raise one very simple question before we delve more into this. So, where do you think Marikina Watershed is located? So, if anyone knows the answer, please feel free to raise your hands. Okay, I think someone's raising their hand. Okay, go ahead.
Marikina Watershed. So, as the name suggests, Marikina po siya.
That's a good guess. Oo nga naman. Kung Marikina Watershed yung name, dapat nasa Marikina siya. But, in actuality, the Marikina Watershed is mainly located at the northeastern part of the province of Rizal, neighboring Manila. In total, it encompasses 4 cities and 3 municipalities. This watershed has undergone excessive increase in urbanized areas, resulting in the degradation of its land. And due to this, mitigation measures such that of a decree proclaiming its upper segment as a protected area, officially naming it Upper Marikina River Basin Protected Landscape in 2011 was enacted to conserve what has been left of it. The implications of urbanization to the watershed has posed a significant threat not only to people but to its stream ecosystems, and this has been proven by several researches that assess the changes in the components of streams that thereby affect aquatic organisms. Actually, a recent study conducted by Peralta and colleagues in 2019 sheds light into such matters. In their study, they highlight how benthic macroinvertebrates can be used as bioindicators of changes in the physiochemical properties of streams, just like what was mentioned earlier. But the question is, why benthic macroinvertebrates? This is because they are one of the most receptive or responsive when it comes to changes in various environmental parameters that are reflected from the health and overall quality of freshwater streams. And all of these are due to their distinctive taxonomic and functional composition. So through these organisms, the researchers were able to comparatively investigate the differences in habitat quality between protected sites and unprotected sites adjacent to it. To give you a brief overview of how they were able to do so, the researchers tested several environmental properties from a total of 16 sites, 8 of which are from disturbed streams, whereas the other 8 are from the undisturbed streams. So presented in the screen is the map representing the sample sites from both protected and unprotected areas. So just by looking at the land cover classifications, which symbol represents the protected sampling sites and which symbol represents the unprotected sampling sites. So again, please feel free to raise your hand. All right, so I think Miss Lourdes, is that you? All right.
Based on the map, I can see that the triangles are located in areas where there is high concentration of built-up. So I'm assuming those are the unprotected sampling sites. And on the other hand, the circles are placed in sites where it's mostly classified as wooded and shrublands. So maybe those are the protected sites.
Oh wow, that's exactly right. Thank you for that. So results obtained from the sample sites are illustrated in this table. The three environmental variables, namely pH, temperature, and flow velocity, do not exhibit apparent variability in the unprotected and protected areas as observed in their values. Though it is worth noting that water temperature from unprotected sites were higher to a limited extent compared to water temperature of the protected site. Though there is only a slight variation in temperature, it may have potentially played a role when it comes to the solubility of oxygen, which is very important. Looking at other parameters such as conductivity, TDS, and salinity, they display evidently high values in unprotected sites, which may be attributed to a great quantity of dissolved and suspended materials derived from agricultural runoff and wastes from settlements. Furthermore, DO concentrations were reported to be lower in these areas because of heavy nutrient loading caused by excessive consumption of oxygen during processes that involve microbial respiration. Another factor that affects low dissolved oxygen in streams is excessive algae growth on account of high amounts of phosphorus. So because of this, hypoxia-sensitive macroinvertebrates are unable to thrive in such habitats. To go further into detail, in terms of benthic macroinvertebrate assemblages, a decline in the diversity of macroinvertebrates can be seen as streams reach the disturbed sites due to the increased amounts of dissolved solids that thereby increases the nutrient concentration as well. In contrast to this, in protected sites, canopy openness, concentrations of conductivity, TDS, and salinity were relatively low. However, taxon richness and evenness of benthic macroinvertebrates are higher and more diverse. This is because of higher concentrations of dissolved oxygen present, attributed to decrease in disturbances such that of domestic and agricultural nutrient loadings. Furthermore, in the protected areas, there was a significant decline in point source pollution, hence the decrease in sewage effluents. This in turn decreased nutrient pollution, giving way for plants and moderate algae growth. Another result generated from this comparative study is a graph depicting principal components analysis, or PCA, and non-metric multidimensional scaling, or NMDS. Through such analysis, the researchers were able to convey that there is indeed a significant difference between protected and unprotected streams, as reflected in the variability of previously discussed environmental parameters and macroinvertebrate composition. So overall, this goes to show that the protection of a particular landscape from excessive human disturbances can effectively restore habitat quality, which can result into the amelioration of the state of both biotic and abiotic components of the environment.
Now that we have seen how anthropogenic stressors such as industrial, agricultural, and residential waste exacerbate these inland waters, it is also interesting to know how the establishment of the freshwater protected area has been an effective way to prevent further degradation in freshwater ecosystems. We can clearly see the significant differences in terms of macroinvertebrate diversity and water quality parameters among the unprotected and protected areas. This necessitates the importance of FPAs as a conservation measure for the eutrophic freshwaters in the Philippines. As mentioned in the Peralta 2019 and 2020, freshwater protected areas is a good initial step for the governments since this serves as a sanctuary for freshwater organisms. The establishment of this should work hand in hand with other conservation measures such as the rehabilitation of riparian zones. With all things discussed, we would like to know what you have learned about trophic status and eutrophication.
I've learned na yung mga downstream areas na nakaka-experience ng different anthropogenic stressors, meron silang high amounts of nutrient concentration, tsaka less macroinvertebrate diversity.
Thank you, Leona. That was an interesting take. How about you, Miss Angel?
Knowing the trophic status can really help us sa pag-create ng mga appropriate management strategies para ma-prevent yung continuous degradation sa freshwater ecosystems natin.
Yes, that is certainly correct.
Hello po. So, ang sagot ko lang po is sobrang helpful po nito since pinapakita niya po ang iba't ibang anthropogenic stressors na nararanasan ng ating mga tropical inland waters. And kaya dapat po gamitin ito ng ating gobyerno as a basis po sana for management and conservation po. Thank you.
Yes, that's right. Thank you. Well, speaking of management strategies, now that we have learned so much about eutrophication, what are the measures or actions we should take as guardians of the environment after seeing such patterns? Well, first, let's sum up what we have discussed. In the San Cristobal Subcatchment, we learned that the river quality in downstream sites that are near the manufacturing plants have high nutrient concentrations and high abundance of pollution-tolerant macroinvertebrates. This clearly indicates that DO level has the most significant impact on benthic macroinvertebrates in streams that receives the industrial effluents. Moving on to the Silang Santa Rosa Watershed, a consistent pattern of high nutrient concentration and lower macroinvertebrate diversity, as well as reduced DO levels, were observed near the built-up and industrial areas. This coincides with the increasing human population density, which confirms that organic pollution is also attributed to anthropogenic sources. Lastly, the interesting Marikina Watershed have revealed a clear distinction between the unprotected and protected streams. The disturbed streams exhibit high suspended materials due to agricultural runoffs and urban waste, indicating high nutrient concentrations and conductivity. It also showed the impacts on benthic macroinvertebrates assemblages, since it exhibits a reduced diversity. This highlights the importance of prioritizing freshwater protected areas, since the study of Peralta et al. 2019 clearly shows its significant differences with unprotected sites. Now that we have all been refreshed from what we have discussed, what can we do now, given that we are in a developing country? First, as the studies have suggested, it is necessary that we improve our pollution control measures. It is not enough that we just stick to simply water cleanup drives. Second, we must provide strict effluent volume control for industrial companies in order to prevent further degradation in these waters. It is also recommended to install more wastewater treatment plants since it has been reported to be successful in restoring fresh water with high nutrients and saving its biodiversity. However, constructions of these requires a huge amount of budget, although there are less expensive ways. One is the riparian forest restoration, which can aid in reducing environmental stressors in our urbanized streams. According to Peralta et al. 2020, we can also adapt other countries' successful restoration projects, since these countries have also had similar problems with us before. Such mitigation efforts by their government include regular stream bioassessment controls and restoration initiatives, which include sediment augmentation, on-site treatment, deculverting, floodplain improvement, and sewage intercepting facilities. We should also use detergents that are not rich in phosphorus, since this is a huge contributing factor to domestic waste, as what we have emphasized a while ago. Such initiatives should work together with establishing freshwater protected areas so that we can have efficient conservation measures. It is crucial to implement strategies in a landscape-scale diversity in the watershed and not just a community-scale diversity, as this can truly improve the ecological and socio-economic aspect. This can truly prevent serious environmental concerns such as eutrophication in our streams. Trophic states indeed help us point out which water bodies need further attention. It is highly encouraged that we should draw our awareness to the importance of using this so that we can prevent further deterioration. Now that we have reached the end, meron kami huling mensahe para sa inyo. Alisin ang green sa tubig tabang natin. Gamitin ang mga aral na to para masagip ang mga tubig natin. Maraming salamat sa pakikinig ninyo. Ito ang aming mga references.